Yes, I think we're online again. I- Hi, hello, and welcome everyone. Microbehunter here. Welcome to another live stream, Microscopy live stream. As always, I'm trying to get all the things sorted out first. Okay, chat seems to be working. Sound is okay. Haha, here we are again. Hi, and welcome again to another Saturday live stream, Microscopy live stream. Today I'm going to look what I got here. I've got a couple of water samples which are collected from my apartment, and some of them contain a couple of bacteria, microorganisms, and I would like to put them under the microscope today with you. And we're going to have a look at what we're able to find here. It's not going to be like a super beautiful specimen, like, wow, look at the colors and all these. No, probably not. But I think it's going to be interesting nevertheless. Okay, so as always, for those of you who are new here, a couple of, how do you call this? Rules of the game, so to say. Please feel free to write comments in the chat or questions. After approximately 1 hour, I would like to answer all off-topic questions, and collectively, so that I'm not getting too distracted. On-topic questions, I'll try to answer right away. Please indicate either at Microbehunter or at Oliver, so it's easier for me to find those questions or comments that are directed to me, because sometimes the community, people from the community would like to talk to each other. As always, it's a tradition. Hello all around the world. Hello from Germany, from Georgia, from the Netherlands, from northern Germany. Estas diable mal varme citie, che anche citie in Austrio estas tre ege mal varme, che mi volas componeble kiel ciu, se meine mi volas anche bonvenigevin. This was a couple of words in Esperanto, one of my hobby languages, because some people here also speak that language. Okay, from Essex, England, from Slovenia, from Belgium, France. Okay, Philippines, Rhode Island, USA, London, USA, Norway. Hello from Wales. Wow, we're almost neighbors. Ireland. Yeah. There is what I will do is every now and then I will. There is already a comment here. Whether I know a certain book. I will after a couple of minutes. What I will do is I will go back to the comments, and I will try to respond to them, because I want to get started right away to simply explain to you a little bit of what I've already prepared today in the afternoon, my time, why I would like to do that. Last week, I put a little bit of food under the microscope. So basically, I put some microorganisms, especially fungi from cheese and so on, under the microscope. These are a fairly safe source. But today, I've got a few samples here that are a little bit, not so appetizing. Okay, but still, I consider them quite interesting. Maybe not quite as I mentioned, as beautiful as maybe some of the other specimens that I have, but nevertheless, hopefully interesting. And I would like to simply look at them with you. A few of them I already looked at, a few of them not yet, so it's going to be also a little bit of an uncertainty for me and myself. The topic today is basically microorganisms that you find in your household, and as a general rule, you should not be growing microorganisms at home. Bacteria, actually not a good idea for health reasons, but we are already growing them without actually knowing it. Or actually, I know that we are growing them. You just have to look at the right places, right? So I collected a couple of samples here from, basically the household. Like pretty much all of you are able to collect those samples as well. Just wash your hands afterwards, okay? I mean, you already know this is the bottom of of a plug. Yeah, and what I'm going to do, however, right now is to very quickly tell you, if you want to find microorganisms, you got to find places that are wet. Yeah, where there is water, there is life. And what I'm going to do right away, the closest source of microorganisms is, you won't believe it. I hope I got enough. Yeah, the mouth, of course. Yeah, there are plenty of bacteria in your mouth, and what I'll have here, yeah, it's a little bit foaming, but what I'll have here as well, of course, also some epithelial cells. But actually, when I did this some time ago, yeah, I found some yeah, some surprisingly large bacteria. Might be different now. Yeah, it depends a little bit on what you have eaten, how long you have eaten something, whether you've had some hot coffee or tea, because hot drinks will actually kill bacteria. Even Anthony van Leeuwenhoek, the first microscopist, realized that when you drink something hot, you're killing off those little animals, bacteria that are essentially found inside your mouth. This is not a good specimen, as you can see, because there's simply too much foam. Yeah, it doesn't matter. Maybe we're still able to see something between the foam bubbles. Let's have a look. Okay, so what I'll be doing is I will always be switching back and forth between my desk and also the microscope. Yeah, and of course lots of bubbles visible, but occasionally we might actually be able to see some bacteria as well. I'm just going up with the magnification here a little bit. I have to, yeah, and let's see what we're able to find. All of these things here are, of course, bubbles, yeah, and yeah, a couple of cells as well. Let's just have a look in phase contrast, and yeah, let's see if we're lucky or not. Yeah. And of course, I can already see them. Just for your information, if you've never seen this before, phase contrast microscopy is very good for observing specimens that are very low in contrast. Bacteria belong to that. And the reason why I want to show this to you is I know some of you know a little bit more about biology than others, but those structures that you see over here, these are my own body cells, epithelial cells that have come off. The little thing, the dot in the center that you see here and here, that is the nucleus. This is where the DNA is. Those large things here are air bubbles, right? But if you look at those tiny structures, especially those long ones over here, these are rod-shaped bacteria. So all of those dark things here, many of them are indeed bacteria. Yeah, maybe some of them are in clusters. Yeah, so this is something that also illustrates a little bit. This is definitely the one over here, right above my head. Yeah, it's a rod-shaped bacterium over here as well. Kind of also illustrates a little bit the difference in size between your own body cells and bacteria, right? So prokaryotes and eukaryotes, bacteria, prokaryotes, they do not have a nucleus, and here these are my body cells that have a nucleus, and that is essentially you don't have to, why am I showing you this as an example here? You don't have to go very far in order to find microorganisms. We carry them around all the time. And as a matter of fact, there are more bacteria growing in our body and on our body than we have body cells. Okay, so yeah, every now and then, in case you're wondering, every now and then when I pause, I'm just looking over here to the side on my screen to simply to look at the chat if there are any questions or comments. So that is essentially those bacteria. Nothing to worry about. They are a normal part of our environment, of our flora, yeah, of our body, and yeah, of course, in the digestive system, they also help us digest the food. The mouth being a moist and a warm environment, plenty of food also available, so an ideal place for microorganisms to grow. Yeah, something I did not try yet. Might try. You can actually also try to scratch some plaque off your teeth, because the plaque, that is a so-called a biofilm that basically contains a lot of bacteria. But I just wanted to show this to you here. And what I'm going to do now is I'm going to switch over to a different contrasting technique, also simply to show you how it looks like. Okay, I'm also going to go up now here. This is DIC. Yeah, and in bright field, I have to change this here as well. Never forget to change the filter. Yeah, that's DIC now. And many of you who have bright field, which is this regular contrasting technique, it will look like this. Yeah, it will be less colorful, but you're still able to see those tiny little things here. And sometimes it's difficult to distinguish, is it a bacterium now, or is it some kind of other fragment, piece of food, whatever. But if you see some kind of a long rod-shaped bacterium, then chances are pretty good that these are bacteria. Right? Actually, I'm a little bit slightly disappointed, because the last time I've done this, I've actually seen some of them actually move around, being motile. I don't know if I'm able to find some of those now. Ah, look at this here. Maybe it's this tiny thing over that you've seen. I don't know if you've caught that. Yeah, these long things that you see are definitely bacteria. That's very typical of them. Huh? But yeah, sometimes they're also moving. Yeah? So you see that the mouth is an ecosystem. Yeah. Also over here, those long one here, the rod-shaped bacteria. Yeah, very easily visible in that sense. Huh? So yeah, just to show you this here a little bit. Yeah, but actually I'd like to see some of them that are actually moving. The streaming motion that you see here is because of the capillary action, because the saliva is spreading beneath the cover glass. Yeah, here again, a long one, a long prokaryote, long bacterium. Yeah, you get the idea. Yeah, air bubbles here. Yeah. So basically, if you want to see bacteria, some people say, "Oh, I want to see bacteria. Can I see bacteria?" Says, well, how can I grow bacteria? But I'm just like, no, you don't want to grow bacteria. Just take a little bit of saliva from your mouth. Okay, yeah. Every now and then, yeah. What about scraping some plaque from my teeth? You know what? Because you're asking. Yeah, I need. Look what I got here. I've got here some. I need to change over a little bit. Yeah, I've got some of these clean tips. I've taken one here, and I'll be using that now to scratch some of the plaque off my teeth. But I'll tell you, I probably don't have a lot right now. I've recently eaten something and had some food, but I don't know if there's much coming off. But let's try it in any case. Between the teeth, where the toothbrush has a problem. And a biofilm. Ah, I got something. Look, this smeary, slimy stuff here. Yeah, that's some plaque. Yeah, good. What you do next is this is fresh tip. You always have to dilute this. So I got some clean water here. Okay, you always want to dilute this with a little bit of water, because otherwise it's too dense and then you're not able to see anything. So let's have a look. Yeah, so just for your info, I'm doing exactly that what Anthony van Leeuwenhoek did back in the 1760s. He looked at everything and he put some plaque under the microscope and he found some bacteria. He called them animalcules, little animals. Obviously, they are not animals, but when people discover something new, you always try to compare it to something what you already know, right? And of course, yeah, you see something moving, then it looks like, maybe these are tiny animals. Okay, so let's have a look here. Again, this is something I've not tried yet. Okay, I have to change now to the scope again. Here we go. Here we go. Oh, that's my plaque. Okay, but actually this does look like there are more bacteria here indeed. Let's go up 40 times. Yup, yes, actually. There is, look, there, look, that's beautiful. Look, there's some stuff moving, tiny things moving around here. They're actively moving. This is not Brownian motion, right? Yeah, so that's actually quite nice. I don't know what this stuff here is. Some food, maybe some biofilm. Lots of bacteria. That's actually much nicer than just the saliva. Look, there's some movement going on here. Yeah, so some motile bacteria. I know what some of you are thinking. Well, that's typically biologists, right? I put my own plaque under the microscope, find bacteria, and I'm super excited about that. Yeah. Some people consider this horribly disgusting and impossible, and yeah, and I think isn't this beautiful? Yeah, I know it's that's a little bit. I look at the world a little bit differently sometimes. Some time ago, I've actually even found some slightly larger cells, but unfortunately not this time. But actually, quite nice to see that there are so many rod-shaped bacteria. So these are the long ones, right? These are all bacteria. And some of them are moving around. Yeah, the mouth is a fascinating place, people. Really, this is, yeah, not surprising because, as I told you, we've got 30 trillion cells in our body. There are more bacteria in, than we have body cells. Look, this guy is moving. Okay. Look, there are lots of them that are moving. Okay, so you get the idea. Okay, you get the idea. Let me quickly, let me quickly look at the comments again. Yeah. He's speaking in tongues. Yeah, I said a few words in Esperanto at the beginning because one of you also commented in Esperanto. It's a constructed language, and a couple of years ago I started to learn it because I wanted to learn a new language. Yeah. Off-topic question. Okay. Yeah, so other than bacteria, what other things are best viewed in phase contrast? I'll quickly answer this. Generally, specimens that are very thin, bacteria are ideal, and individual cells. Generally, water samples, which contain algae and larger organisms and so on, are not so nice in phase contrast because it's very thick and it doesn't look very colorful. I mean, look at this image here. It's mostly black, white, grayscale, not so colorful, right? But bacteria, they can be visible quite well because they are darker. Yeah, here, that's now nice. Yeah, those long rod. Yeah. So actually, phase contrast is ideal for very thin specimens, mostly individual cells and bacteria. Okay. Yeah. So that was basically the request. Some plaque from my teeth. Okay. Yeah. So I'll move on a little bit, but actually I'm quite happy with this. But I do have a couple of other samples that I would like to share with you. We can always come back to those later on. Just last comment. I mean, the equipment that we have nowadays is pretty good, right? Most of us affordable microscopes, even if it's not in phase contrast, right? Then like now, that's a regular bright field. We're able to see a lot. How in the world were the first microscopists with their homemade microscopes able to see those bacteria? I mean, this is incredible. I mean, those guys back a couple hundred years ago, they made their own microscopes, yeah, ground their own lenses, and were also able to see those bacteria. I mean, it's made drawings, obviously, yeah. So it's this is something that still surprises me. This here again is very typical. It's one of my epithelial cells, and here in the center you see the nucleus. Yeah. Look, maybe I don't know how well this actually shows on YouTube when during the live stream, but I do see a whole bunch of tiny moving dots, and they're actively moving. It's not Brownian motion. Yeah. If you have DIC capability, would you also want phase contrast? Depends what you want to look at. I mean, I had the possibility to also connect one phase contrast objective, so I only have one of them, and that's what I've done. Yeah. So where is this? Yeah, so you're back to phase contrast. So you know what? I'm almost getting carried away a little bit with those with this. That's dark field, by the way. Yeah, I'm getting carried away now a little bit with the plaque. Okay, but thank you for the suggestion. I didn't actually think of this, and I'm going to now do the following. I'm going to now show you the first sample that I have here. This is some fungus that I collected from my window. And I'll show you now a quick video on how I collected this today in the afternoon. Yeah, so the window, because of the water that condenses, it's moist. And I was looking around a little bit, and I was actually finding in one corner, generally was fairly clean, but this one window luckily had some stuff growing here on the insulation. And the reason is because the water condenses and then runs down, and it's a moisture is there, right? And this is basically, that's what I've done. Yeah, simply I scratched some of the material off and put it into a little bit of water. Not a video repeats, right? And that's basically where we are. And I said, okay, why not put that under the microscope? And I can already tell you right now, of course, we're able to find a few fungi here. Okay, so so let's do the following. Where is the desk view again? So here it is, and let's take some here, and I always use a new pipette to prevent cross-contamination. And generally, what I would suggest is that you always take a little bit of solid material as well. Okay, so let's take some of this. I don't know if this is enough. Not really. No, you cannot see that. So here we go. So I do want to take a little bit of some solid material as well. And there is always a little bit of luck also involved, because sometimes, There's more, sometimes there's less visible. You drop the cover glass on top and you hope it's reasonably flat, and then we have a look. Okay, and let's go again here to the scope view. No, I don't like this. I want to have the regular bright field view. And let's take this away. At 40 times, we're not able to see a lot here. So this is how it looks like. But we are able to see all of those long string-like structures. And not all of them are actually fungi. There's also some dust, some textile fibers that were caught there. But let's go up with the magnification. I was able to find some conidia in the afternoon today. So conidia are the spores of fungi. But the reason why I also want to show this to you is because I want to simply help you distinguish fungi from other things. So for example, this one over here, is most likely, that's not the hyphae of a fungus. These are not the cells of a fungus. This is mostly a textile fiber. It's, I would say, too irregular, and it looks a little bit like it's twisting. So this is not, doesn't look like a typical fungus. So, okay, but let's move up again a little bit. Here, this is now 40 times. And here we are already able to see something which is a fungus. Do you see this over here? Yeah, these look very much like the spores of a fungus. Also these things here, or the structures that form spores, they are so-called conidia. And that is very typical for fungus. So you see, obviously they are huge compared to bacteria. Maybe I want to open it up a little bit more, just a second. Here, let's make it a little bit brighter. Let's go up 60 times. Oh, okay. See, it's flowing because the... do you actually see this movement? This is because it's already touching the cover glass. Okay, the objective is so... So plenty of spores, and this is the reason why you kind of want to avoid fungi in your apartment, because sometimes those spores are able to spread and they can cause some allergic reactions. So this is so every time when you have somewhere regions that are a little bit colder than other regions, then this is where water is able to condense on the wall. For the movement here is because I'm focusing, okay? So you see the objective is pressing down on the cover glass, and this causes this movement. Let me do it like this. And here we go again. I'm going to do the following. I'm a little bit worried now that I might have made contact with the water. So what I'm going to do now, therefore, is I'm going to take a, I'm going to show you the desk view. So I got some lens paper here, because maybe I got my 60 times objective wet. So I'm going to quickly dry it off. So just have a look what I'm doing here, up there. Right, I'm going in here, and I'm just going to hold it beneath the objective and see if, and indeed, I actually had some, look, I actually did make contact with some water. So what I'm going to do now is therefore the following. I'm going to lower this. Just watch what I'm doing. I'm going to lower the stage. You have to look what I'm doing here at the top. And this one over here is my 60 times objective, and I quickly wanted to dry it off. Okay, but it's already dry now. Okay. So that's because I had too much water on my specimen, and for this reason I just, because I do not want the water to dry on my my objective, because then if there's anything in there, some dust, then it will stick also to the objective lens, and I don't want that. So it doesn't matter. We're back in business again. And have to find the... Here we go. Okay. So here 40 times is going to be good enough for us. If you want to see how this looks in phase contrast again, let's switch it over. I always have to change the filter settings here. Okay. And this is again in phase contrast. And it gives you a slightly different appearance. Yeah. So every now and then, okay, we did this. We all know you did this on purpose to show us how to do it. I don't know if what you're referring to, maybe that I accidentally dipped the, how do you say, the objective into the water. It was not on purpose, but it's something that I'm, how do you say, one has to always expect. Yeah, so it is not something that is unusual. I just also want to say something a little bit. Maybe I might have already mentioned this before, but concerning the cleaning of the objectives. When I bought the microscope, I had a long telephone call with the company who sold it to me. And I asked him about cleaning of the microscope objectives. And this is how delicate is it, and is it dangerous? Am I going to damage it? What happens if I dip the objective into water and cleaning it? And he said the following. He said to me, I shouldn't worry too much about it. Those objectives actually are able to withstand more than actually you might believe, because the front lens of the objective is concave. It actually goes inwards, right? This is also as a means of protection, so that when you touch the slide, essentially the objective itself, the front lens, does not touch the slide because it goes inwards. And he said, therefore, it's a little bit difficult to clean, because the dirt might be inside the front lens, because the front lens goes inwards. And he said what we should do is you should take some of the paper and put it around your fingernail and actually carefully scrape out the dirt from the inside of the objective. Yeah, and he said that many people are actually afraid of doing that because they're worried of damaging the objective. But he said that they can actually withstand more than what you might. I mean, they're designed to be cleaned, right? You don't want to use any hard object, obviously, right? But he said take some of this stuff, this tissue paper, the lens paper, and use your fingernail, put it over your fingernail and scrape out the dirt that has accumulated on the front lens. Yeah? It's quite extreme, what I thought, right? But he actually said that's the way how what you could do, right? I'm saying this just to tell you that apparently those objectives are designed to withstand a little bit more of what you might think, right? So yeah, so let's go up a little bit with the magnifier. So again, this is the stuff from my window, right? And what I have not seen here so much, and is actually the actual cells of the fungus, yeah, which is a little bit of a pity. Because all of this stuff that you see here, those strings here, that's dust. That's not the textile fibers, dust fibers, whatever here. But these are not the cells of fungus. And just if we're lucky, we are able to find a few of them. Sometimes, yeah, here again in the center. Nah, yeah, here. I mean, again, also these are also more like conidia. Yeah. But you see that over here. These are definitely over here. Yeah, the reason is I think I damaged them when I scratched them off. But yeah, all of these things that you see here, yeah, these are all spores, yeah, of fungi. Yeah. But actually, I was able to see is slightly larger. But everything here is broken apart because of I scratched it off. Yeah? You can have pipette tip instead of your nail inside the tissue paper for cleaning more precise. Yeah, if you basically take a pipette tip, yeah, and maybe put a little bit of this tissue paper around it, you might also be, but nothing hard, obviously, right? Yeah. What is this here? Nah, that's a hair. Okay. So this long thing that you see here, that's a very typical either hair or fur or I don't know. And the reason is because it's textured. If you look on the outside here, it's not completely smooth, but it almost looks like it has those scales on it. So very typical for hair. Very typical for hair or fur. Most likely hair, because we don't have any animals here in our house. Yeah, but then again, you never know what's carried in from the outside. Yeah, here maybe this could be, I don't know. Maybe that these things here are actually the cells of a fungus, yeah. Or these here, This here again looks like some hair or dust fiber. And here I see no, yeah. So fungi, plenty of them. Yeah. So okay, I'm just going to move on a little bit. Yeah. Last one is here. You can use a pipette tip. Okay, your camera seems to be slightly out of focus. Yeah, this was a couple of minutes ago. Okay. So, yeah, look. Okay, yeah, I was just reading a few of the comments again. So let's. I always double check every now and then with the time. Half an hour already. Time goes. I still have 1, 2, 3, 4 samples to look at. So let's hurry. No stress, but still let's move ahead because some of them I don't know myself yet. So this was basically sample number 1. So again, this was the stuff that I collected, yeah, from the window here, right? Pretty disgusting actually. It's time to maybe give it another. Yeah, if you want to get rid of these things, I found not hydrochloric acid, hydrogen peroxide to be quite effective. Yeah. So next thing is, let me put this away, I don't spill it. Next thing, okay, what is this here? Yeah, that's one. Okay, let's have another one here. This one I did not look at. So I'm going to show you something which I did not look at yet. It's this sample. Here it is, look. This is the dishwasher. The filter of the dishwasher contains a filter, obviously, and in the filter you find a biofilm. All of this gooey stuff here, that is full of microbes, hopefully. And this is basically what I've done. And I found also this stuff here on the side here. I put some of this as well. I read somewhere, interesting, that how is it possible that those microorganisms are able to survive 90-degree temperature? So indeed, they must have gotten used to the high temperature of a dishwasher. Right? And I would like to see if indeed I'm able to find a few of those. And yes, I do not know what to expect. Okay, so the one from the fungi from the window I did put under the microscope already, but here, the one from the dishwasher, I have no idea. So I really have no idea. So maybe it's going to be interesting, maybe not. Maybe we're able to see something, maybe not. I have no idea. So I need to take again some solid stuff here. It's always also a little bit a question of luck on whether you're able to find something. I think this might be a little bit too much liquid again. And let's drop this on top here. Yeah, you see it's not completely flat. And I'm going to remove some of the excess because I do not want to have too much liquid in here. Because... So let's give it a try, and now I'm also curious on what I'll be able to find. Because again, a dishwasher is quite hot. Yeah. Okay, and of course it might be a little bit difficult to distinguish between cells that might have been part of the food. Okay, I can just tell you a few things that might look a little bit familiar. Those round things here that I see, they do look a little bit like swollen up starch grains from food. Fat droplets generally look much rounder, and look, this is definitely something cellular, but again, it's only, let's go up with the magnification. Let's have a look. And let's see. I would have expected more bacteria actually, or more movement, because yeah, I took so much from the biofilm. But then again, maybe it is indeed the heat of the water that kind of removed many of them. Interesting in any case. I mean, because there is a biofilm there, this looks very much like some kind of a starch grain here. Because of course, it's nearly boiling water, but then again, I mean, one would still expect at least some heat-tolerant bacteria to be there. You see some of the tiny dots that you see here seem to be bouncing around a little bit here, right? But that could be actually Brownian motion. Okay. Do you have iodine close to you to check if this is starch? You bet I have. Thank you for the recommendation. Just a second. Yeah, here's my iodine. Here's my iodine. Yeah, I'm going to check this. Okay, this would be actually quite interesting. I did not expect. Yeah. Let's do the following. I'm quickly going to go into phase contrast again, because if bacteria are present, they're much easier visible. And I mean, no. I mean, of course, look, some of them. I mean, this might be over here. Yeah. But I think you get the idea or the comparison. This is very different than, for example, all of the bacteria that I had in my mouth. In that sense, it's a little bit disappointing, but that's still something where we learn something. Okay? And the only way that I can explain this is indeed it is probably because of the high temperature. Yeah. But let's do the following. Let's or these things here, like, yeah, could be. But you know what? Yeah, what fascinates me now more are those large things here where I have the hypothesis that this is indeed possibly starch from the food. I don't know. Yeah. So what I'm going to do now is I'm going to add a little bit. I'm going to add a little bit of iodine. I have to be careful not to add too much, because iodine is very corrosive. So I have to be careful now that I'm not spilling anything around on my microscope. So the desk view, let's have a look here again. Here it is. And a fresh pipette tip. And where's my iodine? Here it is. Okay. Always careful. Here we go. Okay, I got now some of this stuff on. Yeah. You know what? I'm going to try to lift this, and on the bottom, let's go like this. Any excess I do want to have removed. No iodine. Yeah, no iodine spills, please. Up. So let's have a look now. Here it is. So scope view. Ha! Yeah, sure. Look. Some parts do indeed, but it looks very different again. Where is this? Look, some of these things indeed do stain extremely dark. What is this large thing again? These are some cells. But those large, dark, black things here seem to be indeed starch grains. Okay. Are these fat droplets maybe? Yeah. Here we go. Here more. Yeah, so this indeed does indicate that obviously here, yeah, these are also some cells here. This definitely shows that, yeah, there was plenty of food material in here, but evidently not so many bacteria. Yeah, huge thing here. Yeah, okay, so this basically also has answered a question. So evidently, yeah, there were not quite as many bacteria there as I anticipated. Okay, so let me quickly read a few questions. I'm jumping. Yeah, iodine. Okay. If one day you could get Nile red indicator, yeah, I'll by the way, I've got access now to crystal violet, which can also be used for staining. So I'm trying to build up a little bit a collection of different stains. So, lambda wave plates. I can only tell you very briefly something about this. I don't know much, but maybe later when I'm going to talk about the off-topic questions. So let me see. This was video 1. Was this video 2? Ah, yes, that's another one. Look at this. This is the most, how do you say? Some people say one of the dirtiest things that you're able to find in your household, and that is a sponge, a kitchen sponge. And just look at the color. I mean, this is so, full of bacteria. I remember, even during a long time ago, during one of the first microbiology university sessions, the task was to bring samples from at home, and we had to grow, this was an introductory session, bacteria on Petri dishes, simply by collecting samples that we brought from home, and then in the lab we kind of put them on Petri dishes, and we found out, surprise, the kitchen sponge was full of bacteria, right? So this is nothing new, actually. This is known that indeed there are incredibly many bacteria on a kitchen sponge, and when you wipe your table, all you're doing is you're simply redistributing a lot of bacteria. And I was not disappointed when I actually put this sample under the microscope some time ago, today. And I hope that we're going to be equally successful now in finding a couple of interesting bacteria or microbes, or generally, as a matter of fact, I even found some ciliates, just to, I have no idea how in the world they found their way, into the into the sponge here. But let's give it a try. Maybe we're going to be lucky again to find something interesting. It's always a demonstration problem. Usually when you want to show something, then you're not able to find anything. So, this is now, we have to go up a little bit with the magnification, maybe 40 times. And whoop. And this is basically life in a sponge. Now 60 times. And I think it looks pretty fun. This is, not surprisingly, this is a sponge which is wet. Look at this, even spiral-shaped bacteria. I mean, obviously a sponge that's moist a long time, and where you're wiping things off, large surface area, food present because you're wiping off things. Of course, it's going to be a bioreactor. I mean, so even more than in the mouth. And many of them are actually moving, and actively moving, as you can see. Okay, so this is actually quite nice. Nice under quotation marks. Can I adjust the diaphragm? Yeah, I can go up a little bit more here. Yeah, I can go down with the. Yeah, I can also change the color around a little bit. Yeah, this is DIC now, 60 times, so the magnification is a bit higher. But let's go to phase contrast again. And I have to, as always, I have to kind of. No, this is the phase contrast. Here we go. Yeah, and now you see actually the bacteria again. Darker. Ah, look, a big one. What's this? This is, I don't know. This is way bigger than the bacterium already. Maybe a flagellate or a ciliate. I have no idea how it found its way into the sponge. Yeah. Let me quickly change back again to the, because I have a higher depth of field here. Here it is. Yeah. So life, lots of life. Kitchen sponge is alive. So if you feel a little bit, I don't know, uncomfortable knowing that there are so many bacteria in some kind of a, then what you do is you use simply paper towel and you throw it away. Yeah, then Yeah. But this is actually quite fascinating. Yeah. The biodiversity is incredible. So it's actually it's pretty extreme. So I'll yeah. Please soak your sponges in a water bleach solution for 1 minute before doing the dishes after this. You bet. I mean, as a matter of fact, we're not using the sponge. We're using the how do you call this, the dishwasher, right? But yeah. That's why you don't wanna use a sponge like this for doing your dishes. I mean, look, what shall I say? It's disgusting if we know about it, but somehow humanity still has survived. Yeah. Yeah. So I think there's a huge difference in, look, another one here. There's a huge difference in actually drinking this water. I definitely would not do that, right? And using it for wiping it, and then when the dishes are dry, then many of them are going to die off anyway again. Yeah. Yeah. Oi, that was a huge one. What's this? Ah, look. That's a big one. Yeah. Isn't this fascinating that somehow, yeah, these guys find their way also into this kitchen sponge? They don't just appear on their own, they must come from somewhere. And I'm kind of interested in how are they spread actually over the air, maybe over dust, maybe, yeah? Because tap water generally does not contain them. Or, yeah. Or maybe at a very low concentration, and then basically start to reproduce inside the sponge. Yeah? So it's quite fascinating. Yeah. Yeah, the detergents will, of course, kill a lot of them. This is one of the questions here. However, there's one thing that I just want to say, just because I know some people are getting worried now. Look, these things are part of our natural environment to a certain extent. And even if you kill off 99% of all of them, because you've got billions of them, if you kill off 99%, you still have a couple of million left. Yeah, because the numbers are so high. But that's okay. Yeah? I think what is simply important is that we simply are aware of these things, and that's why knowing this, I'm not using the sponge to clean my dishes. Yeah. So, but again, I mean, yeah, for whatever reason, we humans, are still alive. Why? Because we have an immune system. Right, and being too clean is also not good for the immune system all the time. So there have been studies that children that have been brought up in a highly sterile environment, very clean environment, are much more prone to allergies because their immune system has not learned to deal with these things. And children that have grown up in somewhat environments like farmhouses where there's lots of pollen and so on in the air, right? Their immune system has learned how to deal with these things better, and therefore those people are less prone to allergies. Yeah. So this is also something quite interesting. Yeah. Can ciliates dehydrate like tardigrades and then be dispersed by the wind? Maybe. I can imagine that some ciliates may be able to form cysts. Indeed. Some not. For example, Paramecium is not able to do that. But it can be that maybe microscopic moisture droplets, water droplets, might serve as a transport vehicle. Yeah. So maybe they don't have to dry up completely. Yeah? And again, maybe there is some in very low concentrations. They might be in the water as well. And then when they find a place, so then they will start to reproduce. Yeah. So I bumped myself now. Okay, so what I'm gonna do, what time is it? What time? Okay, 49. So I still have a few things that I would like to show you. So that was sample number 3. I have got sample number 4, which is another one yet, which I want to show you. And it's not so super exciting, a little bit disappointing maybe, because a couple of months ago I was able to find diatoms there, but not this time. So but I'm going to show you in any case. Look what I got here. I'm going to move this up now a little bit because I need more space. How many of you are able to recognize what this is? This is the inside of my coffee machine. Look at this. Look, and where there is water, there is life. And there is this green stuff in there. You might want to clean out your coffee machine every now and then, right? So there's a possibility. I was simply scratching off some of the stuff down here. Large amount of stuff here, and yeah, this is basically what I've done. Yeah, to scratch off. And this one here, it does look a little bit greenish, but it did not receive a lot of light. So yeah, if you were to place it next to the window, then you'd have some diatoms growing in here as well, like I've already extracted from some coffee machines before. And what I've done now is I simply poured these things here. I already got a little bit here in here. And all you have to do is you simply rinse off some of this and you just pour it out. Up. And here we go. Okay. And I will show you something here. Unfortunately, yeah, no, I'm just going to show you. I thought, wow, diatoms, but actually, I don't know. They didn't look quite like diatoms. Okay. Okay, this has got a little stuck. Let's have a look. Make it a little closer again here. Here we go. Oh yeah. It's not quite as flat as it could be. Let's give it a try. And I will put the pipette tip away. Where is my scope? Here it is. I need to again change, and this is. And look what I've seen here. And when I saw this just some before, wow, look at this, all these. Okay, no, this is just the water spray. Look at all these long things here. They look like diatoms, like diatom shells. But then when I zoomed in, actually they didn't. So I want to hear your opinion of this here. Yeah, I mean, there are a couple of things moving around here. Okay, maybe you've seen it. Yeah, but those things here, there's some of them, some life here, occasionally, and I don't know, but those long things here, they don't quite look like diatom shells, yeah. So I think I don't know, some crystals of some sort. I don't know, yeah. So this is a little bit something where I don't really know the answer. Let's go up with the magnification again here. And now you can actually see that, yeah, I don't know. This, it looks, look, some microbes again. But these things here, they don't look like diatoms. I don't know. Huh? Okay, so oxalate crystals. Where would the yes, but where would the oxalate come from? This is a little bit my question that I have, because in this plastic container, I only added water. So I don't know. And then yeah, so it's calcium carbonate maybe. Yeah, which kind of yeah, because of this so-called the calcium carbonate, which kind of crystallized out maybe over the time, but I don't know. It's so that's kind of the interesting thing here a little bit, where I was wow, cool, diatoms, and then essentially no, not really. Wow, what are they? Look at this. What are they doing here? It's kind of funny. Let's zoom in here. I don't know. Look. Yeah. In any case, yeah, even a coffee. Yeah, yeah, yeah. Okay. Yeah, plastic fibers from friction. This was basically the question by scratching it off. Could these be some plastic fibers? I doubt it, because this looks too crystalline for plastic. Because plastic is not really so crystalline, I think. Yeah. But could be. Yeah. And I used a wooden stick to scratch it, so I don't know. It looks like a little bit more like some kind of a deposit, yeah, that kind of formed. Yeah, here again, there's something moving over here. So let's go again phase. Let's go again phase. It actually looks quite nice. Yeah, you see, looks much brighter. Yeah, I wonder what these long things are. If these could also be bacteria, yeah, growing in there. I don't know. Yeah, in any case, quite interesting. Where is the sample from? This sample is from, if you just joined in, the water tank from my coffee machine. Okay, and if you look into here, yeah, if you don't clean it regularly, then you're going to see some calcium carbonate deposit, but sometimes also some discoloration. And if it looks greenish, then these could be algae, right? But this this tank was standing a little bit in the dark, so yeah. And I have seen some coffee containers where indeed there were lots of diatoms, actually quite nice diatoms in there, but not here. So, yeah, the lime scale, something. So yeah, basically the water stains. So I'm going to still move on. I still want to show you 2 more samples. Okay. And also a sample which I did not look at yet. Here, you know this, right? This is pretty disgusting. This is a plug from a sink, the bottom side, containing, of course, biofilm. So it's always moist. Also exposed to high temperature. I also have to say that. But there is still something growing on here, some slime. And this is also a place where there might be something of interest. Or not. We'll see. Yeah. I'll take some. Okay, here. That's too much almost. Yeah, and this also seems to be some kind of hard crystal stuff on here. So, okay, I have a problem now. I'm not able to remove this, so I'm going to show you what you can do if you have a lot of thick material on the slide and you cannot put a cover glass on top. What I recommend that you do is the following. You take a cover glass, and if you were to let's make it bigger. Okay, if you were to drop it on here, it's not going to be flat on here. So what you might do is you take it, you kind of wipe everything off to the side, and you just take the liquid and you drop it over here on the liquid. Yeah. So anything of interest hopefully will be in here. So we're kind of separating a little bit the solid material, whatever it is, from the liquid part here, and I'm going to look at the liquid part, hoping again to find something of interest. Again, this is something where I do not, but I'm kind of worried here that I'm going to dip, the how do you call this, the objective into this here. So I want to get this removed. So look, I'm going to do it like this. So here we go. Okay, I wiped it off. You couldn't see it. Here it is. Yeah. So we go into the microscope view. Again, this is. Where's this? Why is this so bright now? A little bit. I'm a bit confused now. Why is this not? Why am I not getting the full view here? Oh, okay. Now I get it. Here, I have to change this around. So, let me have a look. Also here, actually, surprisingly few things. Also pretty boring specimen. I also expected more life there, and my hypothesis is again maybe because of the high temperature that the plug was exposed to. That maybe this is the reason why I'm not able to see so many living things here. I mean, still gonna be full of it, but not quite as many as I expected. Look at this. So again, some particles here. This is actually quite interesting because usually a biofilm should be quite full of microorganisms, and somehow we're not able to see quite as many as I expected. Here, there might be a few here, bacteria. Those dark dots that you see here are definitely bacteria, but not quite as many as one would anticipate. Thank you very much. Ooh, 2 pages. I got your questions. Yeah. So I'll try to respond to your questions. Yeah. So basically, this biofilm is a little bit, not quite as interesting. However, I do want to show you a slightly more interesting biofilm, where I'm actually working already on another a video. Yeah, so this one from the plug looked promising, but is not quite as exciting as I hoped it to be. There are some bacteria down here. Yeah. But I think the sponge was way more interesting. Okay. So, okay, but I'll move ahead now, and I'm going to show you now some cyanobacteria. Okay, so this is now the next thing. And where is this? And that's going to be now the last specimen that I would like to show you. You're going to ask me, where in the world would I get cyanobacteria from? And as a matter of fact, I've got a water bottle here. Look, I'm going to make this bigger again, taller again. You might know, this is basically because I do not have running water here in my room. I got this water bottle 2 years ago. And look what happened. It started to turn greenish brown down here because on the inside there is something growing. And the only thing that I put in here is fresh water, right? Look what happens when I take out this part here carefully. Look, you see all of this stuff here? Yeah, this slimy green brownish stuff. That is also a biofilm that I would like to put under the microscope. And this contains indeed a few so-called cyanobacteria. So I'm going to scratch this off. You know what? I'm going to put a little bit of water in here. Let's scratch off some of this material here. How do I get it off? Here we go. Look at this slimy, slimy stuff. Yeah. And again, this was just tap water. I only put tap water in here. And over the course of a couple of years, essentially it starts to form this green slimy layer. And that's how it looks like. And when I put it, let's remove any excess here. And these things here seem to be so-called cyanobacteria. And as a matter of fact, they even form oxygen bubbles. So let's go up with the magnification. I have to always adjust the. What you see here is a dense network, and also some moving microorganisms as well. Let's close the condenser a little bit to increase the depth of field. Yeah. And look at this here. This network here, these are not fungi. The reason is I won't be able to show this to you, but some time ago I did a time-lapse video. I'm working on a video for that as well. And because it forms oxygen bubbles. So if you actually wait long enough, you're going to see oxygen bubbles forming and growing. And this is essentially a sign that there is quite a bit of photosynthesis going on. This means that also looking at the size of the cells here, this means that these must be cyanobacteria, which by the way are known to be so-called blue-green algae, they used to call them. But they're not algae, they're actually photosynthetic bacteria. And look, in this water bottle, look, other living things that move as well. Possibly some ciliates or flagellates or whatever. Some other microorganisms as well. So you see that nature is quite resilient, and you can find living things pretty much at the most unexpected places here. Okay, so how do I know that these are cyanobacteria? Let's put it this way, there are not so many options, and strictly speaking, that's a good question. I know it I can infer that, so to say, from the color, from the greenish color, and the fact that they are actually doing photosynthesis. That's number 2. And number 3, from the size of the cell, right? Of course, nature is very diverse, and if you want to be absolutely sure, then you probably should do some kind of a DNA test. Because but simply from the way that they look like, and from the size of the cells, from the fact that it does photosynthesis, because essentially you have oxygen being formed, that indicates that these are most likely cyanobacteria. And also but again, unless these are very, very small eukaryotic cells. And cyanobacteria are quite common. And as a matter of fact, I've seen sometimes also that some of them actually move, just like oscillatoria also moves, but I have to find some kind of a place where some of these cells have actually broken off. And then some of those filaments actually also were able to move slightly. You can see that also in time lapse better. Maybe this one, One have to wait a little bit and be very patient, and then you can actually see them. Some of them move slowly and glide slowly. Okay, let me quickly. Domestic hot water is way more dirty compared to cold water. I also heard that. I think it depends a little bit on where the water has been heated, whether you've got a heating system. But I also heard that essentially that I can imagine maybe that hot water maybe dissolves some kind of maybe more substance, or if it's not completely hot and warm, this can also promote, of course, bacterial growth. So what kind of network is this? These are most likely, I have to be careful what I say, probably so-called cyanobacteria, which are found in a biofilm in this water bottle here, right? And this is, by the way, that's another reason why when you have drinking water bottles, right, which you always refill, many of them actually come with a cover. And people think, well, this is simply to protect the bottle from breaking, but in reality, it protects the bottle from light. Because if light is able to reach the water over the course of a long time, actually you have a little ecosystem growing in there. Okay. Wherever there is energy, for example, that's also a comment here, wherever there's energy and maybe water, there can be life. And the energy in this case would be in either solar energy or organic material, right? So an energy source indeed, and water. These fibers are made of cyanobacteria. Again, the question is, how do I know that these are cyanobacteria? I suppose that they are, simply from the way that they look like, and it's not surprising either, because you find them all over the place. So this is not a huge discovery. And again, here I cannot show this to you now because we don't have the time. But I'm working. Here is an This is what you see over here. This is a bubble, right? And what we now, if we wait for a long time, then we're actually able to see that this bubble will grow. What I can do is while I read through the questions, I will put the arrow right at the edge of this bubble here. And then we can actually use this. I'm going to leave it there. And then maybe we can see that the bubble is actually growing because the cyanobacteria, what they're doing is they're doing photosynthesis and they're producing oxygen gas, right? And then the bubble should be growing. I also want to tell you the following. Just if you want to repeat this experiment, you have to be careful because the size of the bubble also seems to be changing when the water evaporates, and when the thickness of the water between the cover glass and the microscope slide, when it changes, then this might also give the appearance of the bubble size changing. but I think you can see already over here that the growth is pretty rapidly going on. Yeah, because the arrow, look, is now in the middle of the bubble, and it was just a minute ago it was on the side. Huh? So you know what? I'm gonna move the arrow again to the edge of the bubble. Okay, this arrow I'm gonna move to the edge of the bubble as a reference, and then let's the tip of the, and then let's wait again and let's see if it grows. Yeah. Yeah. So I'm going to use that time now to maybe have it. We're just gonna leave it like this, and I'm going to try to read off or go through a couple of your questions. Okay. What time is it? Oh, it's 1:10 already. Time passes. Do you know the book? Yeah, I'm not showing you the sheet now. I'm just reading this off here. Do you know the book? Walter Dioni, Safe Microscopic Techniques for Amateurs, Slide Mounting. Thank you for the recommendation. I do not know this book. Lots of old formulas done with safe ingredients instead. Thank you for the recommendation. You know what? I'm going to try to get the book. Yeah. No, I don't know the book, but it's thank you. Off-topic questions other than bacteria. What other things are best viewed in phase contrast? I briefly mentioned this already. Phase contrast is probably best for those organisms that are very small and thin, so individual cells, for example. Thicker samples, like water samples with a lot of algae in it and water microorganisms, generally they don't look quite as nice. It looks a little bit, yeah. So I would say phase contrast, bacteria and single-celled microorganisms are pretty good in phase contrast. But other than that, for casual observation, I probably think it's not worth getting it, because phase contrast changes the natural colors around, and then you don't, yeah, you see it kind of, yeah. Look, the bubble is really expanding. Can you try looking at, I don't know this, didiniums. Sorry, I don't know what this is. I'll try to find this out and then I'll report back. Yeah. Scraping the plaque from the tooth. We've done this. Found some interesting microbes. Off topic. Anyone seen BI on eyepieces? It seems to give a clearer view than just a wide field only. Is it a magnification? No. BI, I don't know. I've not seen this on eyepieces yet, but the BI does not refer to the magnification. Okay. I wouldn't know what this means. Yeah, because the magnification of eyepieces generally in most cases is 10 times. Yeah. Okay, you can use a pipette tip instead of your nail, basically for cleaning the objectives. Then somebody said you got to be careful with that a little bit. Okay, a minute ago I noticed I had already an old chunk of ginger root. Lol, I watched some of the really long wiggly animals under 1,000 times. One had an oval creature stuck on its head on one end. Yeah, generally, if you you find old decomposing material or so, plenty of things to observe. Do you have any iodine? I tried that. Okay. Use Löffler to find DNA in bacteria. Yes, Löffler's solution is methylene blue. And it will stain bacteria as well. You have to heat fix them, and I think maybe not necessary to heat fix them, but it's usually done. And I'm also going to try to get some crystal violet, which can also be used to stain bacteria. If one day you should get Nile red indicator, I will also, yeah, I'm currently working on getting more stains as well. Can you tell something about lambda wave plates? Lambda wave plates are used sometimes in polarization microscopy, and what they do is to convert so-called linear polarized light to circular polarized light. They can basically give you more colors. That's basically the bottom line. How it works from a physical perspective, I don't know. But essentially lambda plates are frequently used to change the colors around in polarization microscopy. So I cannot really tell you a lot more than that because I don't know much more. Yeah? Just thought about soaking our toothbrush in water for 5 minutes and taking a sample. Yeah, look, the bubble is moving. Yeah. So that is indeed a possibility as well. Now two bubbles joined together. Oh, there's another. It seems to be another bubble. Yeah, let's. So I switched. I went down a little bit with the magnification. Let's move it here again. Yeah, and please soak your sponges in water, bleach solution. Yes, essentially, bleach or in hot water, or best yet, don't keep it wet. That's the bottom line. If it's always wet and never dry, then essentially you're growing your bacteria there. Yeah? I stopped using sponges years ago because I suspected they were gross. Yeah, but then again, yeah, lots of people are using them, and we're still doing fine. Our immune system is remarkable. Yeah? Can you make a video one day about DIC? What components it's using and how everything works in details? There's not much info on YouTube about that. That's correct. However, it depends. Upgrading your microscope to DIC generally in 99% of the cases is not possible because it's very company specific. So if you're interested in getting DIC, yeah, you're basically, you cannot simply upgrade it like that. You need to get components that essentially, yeah, the company designed, right? And there are not so many companies that actually make DIC equipment. It's usually the high-end microscope manufacturers. I also know that the company Euromex in the Netherlands actually also has a DIC microscope. Yeah? And some people are also using, actually used more older microscopes, and they're buying the parts together, and that's also possible, but that might be a little bit more difficult to get the parts. On a serious note, human body is made of to deal with these bacteria on a daily basis for the last millions of years. Yes, of course. That's why we're still around. And as I said, our human body has more bacterial cells growing on us and in us than we have body cells. So that's actually quite fascinating. We're actually from a cell count made more of cells that are not part of our body. But actually, now we could actually consider them to be part of our body because, yeah, they're part of the normal environment. And we need them also for digesting food and so on. Yeah? How do they get there to reproduce? They need to be critical minimum number to do so. I don't know exactly now. So the question is, how do they get there, the microbes, to reproduce? There needs to be a critical minimum number to do so. I don't know exactly now to which specimen you refer to, but actually, if the conditions are right, you do not need to have a critical minimum number. One bacterium is enough. If the conditions are right, this one bacterium will divide, and then there are going to be many of them. Yeah. So let's put it this way: for animal populations to sustain themselves, I don't know. You have a population of elephants somewhere, right? They need to have a certain genetic diversity in order to survive as a population. Otherwise, they're not able to survive on long-term and changing environment. So for higher animals, this is really important to have a certain minimum number for the population to survive. But for bacteria, it's different because they reproduce by cell division, and one bacterium is enough, and if the conditions are right, it will reproduce. Can ciliates dehydrate like tardigrades and then be dispersed by the wind? Maybe some of them can. It depends if they're able to form cysts or not. I don't know which ones are. Yeah? I have a question. I invented a new Nikon with a folded tube. I sold my Nikon E200 to work since I was tired of using a broken one. Ah, okay. Was it a mistake? I'm sorry, I'm not able to. Let me quickly show you this question here. Sorry, I'm not able to answer this question here. I invested, not invented, in a new Nikon folded tube. I sold my Nikon to work since cannot answer this question. Sorry, it depends a little bit. Yeah. Know too little about those microscopes. And honestly, mistake. Look, ultimately for most of us, this is a hobby, right? And whatever we do, we have to enjoy what we're doing, right? And what's right for one person might not be suitable for another person. Yeah? Oxalate crystals, this was referring to the crystals, or what we think of being crystals, yeah, in the coffee container. Calcium carbonate crystals, maybe, I don't know. Do you have hard water in your area? Yes, the water in our area is very hard. Hard water means high in calcium carbonate. And indeed, yeah. And this could be the case, yeah. If you suspect it's carbonate related, you could try to dissolve it with vinegar. Yes, this acid generally will try to dissolve it. A drop for hydrogen peroxide to see if it's organic. That's also a possibility. That's a good one. Hydrogen peroxide breaks down organic materials, and essentially if it's inorganic, it will not be affected by hydrogen peroxide. Yeah. You breached 100 viewers mark. Really? That's cool. Thank you. Yeah, and oil immersion will be loaded with bacteria. So, what I'm going to do now is the following. For the next, so thank you for your questions here. Okay, so if you're new to this format, these are questions that essentially my son helped print out, which you were posted during this live stream here. At the end of in a couple of minutes, I'm going to go through the remaining live stream questions again, and in case I was not able to answer some of them, where is it? For those of you who just joined in, this here, look at the bubble, yeah? This kind of shows there is a lot of photos. Look, more bubbles. Isn't this nice? Look, yeah. So lots of photosynthesis happening here, yeah, in this biofilm of what I believe to be cyanobacteria. Yeah. So these bubbles were not present before. Those cyanobacteria produce oxygen like crazy. As a matter of fact, they produce, I don't know, 20% of the oxygen in our atmosphere are produced by those. By cyanobacteria, something like that. I don't know if the percentage is right. In any case, a significant amount, right? And for those who just joined, I got it from this water bottle. On the inside, there is this layer forming. Yeah, it looks a little bit greenish. Yeah, so this is actually a sign that there is something growing in here. Huh? So, and what I'm going to do now is the following. There are a couple of off-topic questions or some questions. And I'm going to quickly go through it because some of you asked something and I'm going to skip ahead and only to those where I actually see my name here. A job for hydrogen peroxide. Okay. Okay, warning, some folks forgot to leave a thumbs up. Thank you for reminding. Okay, I just skip ahead. Off-topic question. Clearly, many Microbehunter takes an interest in your microscopy hobby, my son. But what does Mrs. Microbehunter think of all of this? She tolerates it. Yeah, it's the point. I'll give you. Okay. Look, serious. I'm a biology teacher. I've been a biology teacher now 26 years. Teaching middle and high school biology. I've got a degree in microbiology as well. And a couple of years ago, I started 7 years ago, actually, I did an anniversary 7 years video just a few days ago. I started this channel, now some serious talk here, because when we do microbiology science on an academic level, at university or in school, it doesn't matter. There is always the danger that things can become very theoretical and very dry very quickly. Okay, this is simply in the nature of academics. Even in schools, of course, we try to make school lessons interesting, but there is, with, I don't know, 20 students, 30 students, there are only a limited number of things that you can do. There's always the danger that the initial interest that people have in a field, they stop losing interest once they start to study it on a more academic level, right? We have as teachers, educators, we have as parents, we have to be careful that the natural interest that children have, adults have, is not destroyed by an academic approach. And I decided a couple of years ago, therefore, I'm missing the words, to recapture a little bit the wonder of the natural world. I studied biology because I like to observe nature, I like science, I like observing, I like to be surprised by things. And sometimes when you study things on an academic level, the natural curiosity becomes lost. And we as science educators, as teachers, as parents, whatever, as hobbyists, we have to make sure that this natural wonder of the natural world is, that we don't lose that. So just some serious words here. Yeah, off topic. Yeah, domestic hot water is way more dirty. Yes, and I can understand cyanobacteria growing in there, but what carbon source are there other organisms using? They must have been eating the cyanobacteria. Correct. It works like this. What we have is we have a mini microscopic food chain. So do you see those? Where's this? occasionally I just saw some, okay, look, there's some stuff floating around here, swimming around. Yeah. Many of them are heterotrophs, are feeding on organic material. Where does the organic material come from? Well, the cyanobacteria made them from carbon dioxide, water, and sunlight by photosynthesis. So those cyanobacteria are the beginning of the food chain. Yeah, and then essentially other organisms are able to feed on that. Yeah, it is a food chain what we've established, a very small one for sure, right? But that is essentially, something what you find in ecology. You always have to have photosynthetic microorganisms at the beginning, because these are the only ones that are able to convert the energy of the sunlight to the energy of organic material. Yeah, and then other organisms will feed on the plants or the cyanobacteria. Yeah? So it's the start of a food chain what we have here. So that's a central question here. Yeah. So what kind of network is this? If you just join, these are basically most likely cyanobacteria. Okay. Can you zoom in a little more, please? You know what? Yeah, why not? So I'm going to use the 60 times, and it's too dark. Here we go. Here it is again, one of those bubbles. Usually you see it better on the edges. Here it is. And then, I don't know, sometimes I was able to actually see some of them move, actively move, the cyanobacteria themselves. But here's a nice network. Yeah. So, and Wikipedia is a genus of unicellular ciliates with at least 10 accepted species. All are free living. Thank you. Okay. Somebody joined in. Hello from RI, Rhode Island, I suppose. Are the concave slides good? There are these slides that have a little round hole. I will show you. It's an off-topic question, but one that's actually relevant as well. Look what I got here. Just because you're asking, and then I'm going to call it quits very soon. Where is the desk view here? So the question is those slides, and you see that they have an indentation, a little bit, they're concave. They're a little more expensive. And indeed, they have their use. For example, if you want to observe some, let's say, water fleas, water crustaceans, then basically you can put them in here and you put a cover glass on top and they do not become crushed. If you want to observe smaller organisms, like those cyanobacteria, microorganisms and so on, no, it's not good, because the water would be too thick. Yeah, and then they're going to float vertically, and then it's always out of focus. But for larger specimens, you can definitely use them, especially if there is the danger of them becoming crushed. So what I do is because they're a little more expensive, those slides, I of course always clean them and wash them. Yeah, but that's basically the question about those concave slides. Yeah, so I have a few of them that I use, but not on a regular basis. Only it depends on the specimen. Yeah. So, there is an answer here to that. At 31 minutes past, off-topic question I asked that came after you've received. Off-topic question I asked that came after. Yes, I maybe, I don't know, did I skip your question? I don't know. Jesse was asking. Okay. And I don't know if I didn't miss the question or not. Off topic, Dave, can you do a chat about idioblasts at some point? I'm trying to learn more about them for succulent plants. Yes, as a matter of fact, I have somebody some months ago sent me some beautiful cross sections of water lilies, and they contained idioblasts. Idioblasts are crystals that are formed inside cells. Yeah. And I did show them maybe several months ago. I don't even know in which live stream I showed them. But indeed, I can show them again. Okay, so idioblasts are cells that are basically contain crystals, and sometimes water lilies basically form those crystals to collect them as waste products, so to say. Yeah? So very quickly, I'm scanning through the comments here. Yeah. Okay, ignored. You already answered it. Thank you. Okay. Okay. Yeah. So what I'm gonna do now is, folks, I think it's enough for today again. For those of you who joined in late, all of this stuff video will of course be available online. Just as a short summary, today I put some samples from my household under the microscope, usually some biofilms, and I found out something interesting. Not surprising though, is that some biofilms from the dishwasher and from a plug over here that I have, where I expected to find a lot of bacteria, actually didn't quite contain as many of them as I expected. And my hypothesis is that maybe it's because it's also exposed frequently to hot water. Yeah, I don't know, but that could be an indicator. And the worst sample, worst under quotation marks, is the wet sponge, which was full of life and contained many bacteria. Okay, yeah, and I think I'm going to still another comment here. Do you still go back to your journals or drawings sometimes? I haven't been drawing now for quite some time. As many years ago, I actually started to make some drawings of microscopic specimens that I found. This was over 20 years ago already, but have not been doing so. Yeah, and I've basically also, because a little bit of lack of time as well, so I decided to place my focus now on videos and video streaming and things like this that I'm doing right now. Yeah, and I gave you some nice video and live suggestions. Yes, thank you very much. I've read them. Alexandre commented that, and I'm going to pick up on that. Okay, and so thank you very much. And I'm going to call it a day. For today. I wish you again all the best. Happy microbe hunting, and hope to see you again next week. And I'm as always uploading also some regular videos, so you can also of course check those as well. Okay, all the best, and bye bye.